So we're going to start off with Yann Surget-Groba coming to tell us about conservation genetics of Blanding's turtles at the northern edge of its distribution range.
So I'm Yann Surget-Groba. I'm professor at the Université du Québec en Outaouais in Gatineau, Quebec. And today I'm going to talk about a project. So it's more a project. I won't show you much result. It's a project that started recently on the conservation genetic of Blanding's turtle at the northern edge of its distribution range. So first, the distribution of Blanding turtles. So the main distribution is centered on the Great Lakes, with some disjunct population. So in Canada, we recognize two main populations. So the main range, which is the Great Lakes St. Lawrence population, and an isolated population in Nova Scotia. So both these populations are considered as endangered at the federal and provincial level. And actually, the whole species is classified as endangered by the IUCN. So we consider that the population of the Great Lakes, which is the one I'm going to talk about today, the size has decreased by 60%. It's an estimation. We don't know much about the population size, but apparently the population is declining. So that's why it was classified as endangered. So there's been a recovery strategy that was published a couple of years ago by the federal government. And this recovery strategy has as a long-term objective, which is a 50-year objective, to increase the abundance, maintain, and if possible, increase the area of occupancy. And also the aim is to ensure the viability of the local populations. So we have also at the Quebec, in the Quebec province, Plan de rétablissement has been published last year. And also I just put you here two objectives. There are 5 different ones, but objective 3 is to basically know better the distribution of the species and the habitat, critical habitat of the species. And the 4th one is also to evaluate the viability of populations. So we can see that both recovery plans insist on the distribution and the viability. So we need actually to understand better the distribution and mainly the abundance of population, because a lot of records are historical, and we're not sure if the occurrences are still exist today. So we need to identify the critical habitat of the species so we can protect it, and we need to estimate the population viability. So that's the main 3 points where data are lacking, and we need to have more information to help the recovery strategies. So how do we infer or estimate the distribution and abundance of the species? So mainly on these species are 2 main approaches, so which is visual survey. So we just go in the field when it's nice and sunny, and we try to see turtles that are basking. Or if we want to have more precise estimation of the number of individuals, we're going to do some trapping. So here, that's the field work we did this spring, where we put hoop nets, and we go every day for, in this case, it was 9 days in a row to see what we can catch and estimate the number of individuals in the population. So to estimate the population viability, we need to know the population size, because there are a lot of occurrences basically where we just have 1 individual. So we don't know if it's really a real population. Maybe it's just individuals that were released. There are a lot of individuals that are captive of this species. People like to take them and bring them at home. And sometimes they escape. So we can find individuals in weird places. And actually, they are escaped animals. They are not a real population. So we need to know more about the population size, the trend also, are the population increasing or decreasing in size? And we need to know also what is often neglected, but we need information about the genetic diversity. When we do model on population viability, we need to understand the genetic diversity, because population with low diversity will have a higher extinction risk. And we need to know also the connectivity between populations. So here is just an example of occurrences that we have of the blanding species. So we have a lot of areas of occupancy, but these areas are disconnected one of the others. So we need to understand if the individuals are going to move from one area of occupancy to the other, or if they stay in the habitat. So that's really important, because in this example, let's say we have 1,000 individuals across the whole area. If all the population are connected, that means we have one population of 1,000 individuals, which is okay. But if all these little sites are disconnected, that means that each of the area is actually much smaller in size, and they evolve separately, so that we can have problems of inbreeding and that increase extinction risk. So that's very important information to get. And again, for that, we need to trap the individuals to estimate the population size and to collect tissue sample to be able to do genetic analysis. So, So all these methods are very expensive in time and human resources. So that's very difficult to get this data. So for instance, the field season we did this year, this spring, we went for a total of 32 days of field work with many people in the field. So in total, it was 166 person days. And we followed 8 different sites, and we captured about 187 turtles in this time. So that's many people involved, and that's very expensive in terms of time and salary of the people that are in the field. So we try to see if there would be alternative methods that are cheaper, faster, and at least as efficient as the traditional method. And one method that is used more and more, that is very popular these days, is the study of environmental DNA. So environmental DNA, or eDNA, is DNA of a species. So that's the genetic material of one species that is released in the environment. So it can be released through the skin, the feces, the urine, in all these in all this biological material, you have DNA, and the DNA goes in the environment, and we can use it for different things. So first, we can use it for species detection. So for instance, we can take water and try to see if we can find DNA of the blanding turtles. Because we can measure the concentration of DNA, we can also try to see if we could estimate the species abundance. And lastly, because we can look at what is the DNA that is present, we can try to see if we could estimate the genetic diversity directly from the water. So species detection is used very currently. So now it's well established that it's a very efficient method to detect species, especially cryptic species that are difficult to see by other methods. But species abundance and genetic diversity are still very experimental. So how do we analyze eDNA? So the first step is to collect water samples. So here's the setup we're using. So basically we have a drill that is going to drive a peristaltic pump head. And we have this setup, so that's a filter holder. So we first have a pipe that is going to take the water. Then the water is going to go through a filter that is in this little holder here. And then the water is going to go through this tubing, and we can measure the quantity of water filtered here. So that's the setup we're using to collect the water. So usually we try to filter about 2 liters of water at each location site. Then we're going to analyze try to purify the DNA that is present on the filter. So we're going to use what we call a quantitative PCR. So nowadays, most people are aware what is a PCR test thanks to the COVID. So basically, we're using exactly the same technique. Instead of trying to detect viral DNA, we try to detect turtle DNA. So we use this machine. And the result we have is like that. So basically, each line corresponds to one sample. So the blue one is standard of different concentration, and the pink one are the samples we're trying to see. And basically, if there is a line, that means that we detected the DNA of the species. So that's for the detection. But because we have standards of different concentration, we can do a standard curve. So here the points, the round points correspond to the blue, the standards. And then we're going to put our samples here, and we can estimate the concentration of DNA that is present in the sample. So another possibility is to sequence the DNA. So here we just try to see if we have DNA of one species, but we can also sequence what we call a barcode. So that's example of different DNA barcodes. So here in more detail, you have the sequence of 2 different species. And if we have the sequence, you can identify the species. But each species will have different sequence. So here the stars are different between the 2 species. So by sequencing, we can estimate what species are present. By, we can detect many species at the same time. And within species, we can also measure genetic diversity. So based on this observation, we launched a project on blending turtle conservation genetic. And it's a project involving many people, the National Capital Commission, the Ministry of Forest, Fauna and Parks in Quebec, Nature Conservancy Canada, and Environment and Climate Change Canada. And we want to answer different questions. So can we use eDNA to detect blending turtles? Can we use eDNA to estimate population size? And can we estimate population genetic parameters, so diversity, connectivity, directly from eDNA? So for the first point, so we can answer yes, we can detect the turtle eDNA. So that's just a detectability analysis we did. So here we have the number of samples. So usually we collect up to 30 samples in one environment. And here that's the replicates of the reaction we do. And basically, the more replicates, the more we have a good probability to detect the species. And now, so we're sampling 15 sites, and we're doing 4 replicates, and we have a 100% of detecting the species if it's present. So the second and third points, I'm not going to go into that because Laura and Daphne are going to present their project. It's just starting now. So they're going to present their project a bit later. So many people help for this project, so for funding and in the field. So I'm not going to cite everybody, it will take too much time, but all these people help in the field, and most of the photos that were in the presentation are from Yohann in the Ministry of Forest, Fauna and Park of Quebec. So thank you very much, and if you have questions, I'll be happy to answer.
All right, we're going to hop over to Damien Mullin, who's going to present his talk called Using Science to Inform Species at Risk Management, Predicting the Effect of Forest Harvest on Wood Turtle Habitat Suitability.
So the wood turtle is a threatened species here in Canada. This is a range. It's quite just disjunct throughout here. It's not as homogeneous as the range map shows. And they are declining throughout this range. they have multiple threats, but one threat to wood turtles is forestry. And forestry is a widespread threat, so it occurs in approximately 40% of their Canadian range, but is a data deficient threat. So for those of you that don't know wood turtle biology, you may be asking why are wood turtles affected by forestry? So it's because of their spatial behavior. So the males and the juveniles typically stay close to the river, while the females move like perpendicular to the rivers during the summer. And we've had females move up to 1.3 kilometers perpendicular to the river into these forested habitats. We don't know why the wood turtles move into the forest or why they choose forested habitats. There may be thermal benefits. There may be food benefits in there, and maybe partitioning of food resources between sexes. And this is actually something we're looking at with a senior honors student right now doing some stable isotope work. It can also be females avoiding males, or possibly even females avoiding predators. So to protect wood turtles, in December, there was critical habitat defined at the federal level, and this is a 200-meter buffer. And some important things to know is that this 200-meter buffer is not like a whole buffer. It excludes coniferous forest. So if you have like a black spruce stand or something, that would not be included in the critical habitat. And it's also important to note that this is a federal critical habitat designation, so it only applies on federal land. So you're thinking military bases and national parks. And there's no forest harvest allowed within here. Well, there is 1 hectare, but that's not really a harvest block size that's feasible. So the critical habitat guidelines are a bit contentious when we move to provincial levels, and they're unlikely to be adopted provincially in New Brunswick. Currently, they have 30-meter watercourse buffers, and there's already pushback from industry on this. So if you move a 30-meter buffer to 200, when wood turtles are thought to be widespread throughout the province, you're not going to have great voluntary compliance with the species at risk act. And there's also no studies that have extensively shown forestry degrading wood turtle habitat. So there's always going to be this pushback of where's the science that's showing what forestry is doing to wood turtle habitat. And there's also thought that some aspect of forest harvest may provide beneficial indirect effects for wood turtles, such as creating thermally optimum openings in the canopy and early successional food sources. So our project is looking to predict the effects of forest harvest in wood turtle habitat suitability. And we're doing this using a method called using a resource selection function to model habitat suitability. So the first step of this is having a viable source population. And on these, so we have GPS loggers on turtles. We're using remotely sensed data, using LiDAR to look at canopy and shrub density, and enhanced forest inventory data to look at stand level data to see what kind of stands the turtles are using. We also have a lot of ground truth data. We have food densities. We've done a lot of slug surveys, some predator densities, and then we're going to apply this resource selection function. The second step to our project is to collect the same habitat variables at chronal sequence clear-cut harvest blocks, age 1, 5, 10, 15, 25 years, and late successional forests to look at how wood turtle resources change over time. And for those of you who haven't seen a harvest block grow up, at the bottom of the screen right now is a block that was cut in 2009, and this is a block 3 years later. So you have that shrub layer, and in terms of wood turtle microclimate, it's actually quite cold once you get below the shrub layer. So resource selection functions are becoming quite popular in the literature. Here's one that was used for a desert lizard. On the y-axis here, we have use, and as we see shrub cover goes up, use goes up. So then you can make kind of like a composite images of multiple rasters and put these all in. It's very similar to Maxent models that we've seen. So we have some preliminary results that we could show just from our wild turtle data. So here we have a map of canopy density. In green, we have closed forest, and in small little orange spots, I don't know if you can see my mouse, that's little pieces of open canopy. We seem to have turtles moving towards these areas of open canopy. And also if we move to maybe some stand level data, some EFI data, we do see lots of wood turtles using these softwood or coniferous stands, it's forestry talk, using these coniferous forests. So we are hoping to redefine some stuff in the critical habitat designation. And with that, we're hoping to better delineate wood turtle critical habitat and improve conservation efforts by using evidence-based, data-driven strategies. So thank you very much, and I don't think I have time to go over these knowledge. Thanks, everybody.
Yeah, so we're going to now jump over to Matthew Keevil, who's going to talk to us about a model of seasonal variation in somatic growth rates applied to painted turtles and snapping turtles.
Okay, so today, you know, kind of late on a Sunday afternoon, it's time to get into some statistical models, so some growth modeling. So I hope everybody's ready for that. What I'm going to talk today about is trying to develop a seasonal model to incorporate into von Bertalanffy growth curves for temperate ectotherms that experience a seasonal climate. And the species that we're looking at for this are painted and snapping turtles, and they are studied at the Algonquin Wildlife Research Station in Algonquin Provincial Park, Ontario. And as many of you know, this is a site of long-term life history studies for these species. So it's been going on for painted turtles since 1978, and for snapping turtles since 1972. And of course, this is a cold climate, especially because it's near the northern range limit and it's also a slightly higher elevation. So it's very strongly seasonal. So why do we want to look at growth? It's because it's an important component of life history. So on this plot, what I've shown here is just a von Bertalanffy curve for snapping turtles in Algonquin. And so it shows female median age of maturity. So that's the sort of life history or like vital rate parameter that you would want to estimate that growth models are good for. And also it lets us analyze things like sexual size dimorphism, which you can also see here with males being larger than females. And when we model growth, what we're modeling is size as a function of time. And depending on your data, there's different parameterizations that you could use. So you could, if you know the age of the animals, you can use size at age parameterizations. And if you don't, you can use the interval or recapture parameterization. So that's change in size for a unit change in time based on how far apart the observations were in time. But as you can see from this plot, so I'm just showing an example, one male snapping turtle over about 17 years. And you can see colors indicate that it's captured and measured at different times in the growing season in each year, just whenever it happened to be caught. And this is a challenge because, of course, growth is not equal at all points in the season. And so it can make it difficult to really quantify time. And in order to illustrate how that's typically done, or at least some common ways that it's done, I'm going to use this kind of conceptual diagram. I'm going to call the data analysis sausage. So we're trying to make delicious sausage. And the first thing we do is you take all your observations and you put them in the data processing machine, which for most of us is Microsoft Excel or similar software. And it's this processed data that's then fed into the statistical model. And it's at that point, most of the attention is paid by researchers, collaborators, reviewers. You know, they look at the data going in the statistical model. And sometimes there's things that are important that occur at the processing stage, but are kind of invisible. We don't really think about them as much. So what I want to do is make some of those implicit things explicit, particularly time. And so let's look at an implicit model and how that would look if you actually plotted it out as if it was part of the statistical model. So a common approach is the active season implicit model. And that's basically just defining time as the number of active season days between two observations or since birth. And so what I've plotted there's kind of two plots. One is rate, so it's the relative growth rate over time, and the other is accumulation, so the accumulation of size over time. And what you can see is that on rate, there's constant growth rate throughout the active season. It's always the same until, except outside of the active season when it's 0. When you look at the accumulation, you can see there's these abrupt transitions at the start and end of the active season. And so there's certain implied assumptions. One is that the growing season is the same as the active season, that some of you might be able to think of reasons why that might not be true in real life. And also growth is uniform in the active season. And particularly because it's an implicit model, that the active season timing is known without error and it doesn't change. And of course, we can also think of that might not be true. Some other things about the active season implicit model is that it implies two parameters. So it takes two variables to describe this. So either a start and an end, or a start and a duration. And it requires independent knowledge of growth timing. So it's based on some, just our knowledge and natural history of the particular study population, because it's not responsive. It's not based on the data that we're analyzing. It's set ahead of time. And then just to quickly go over another approach, which is fairly commonly used, is just the ignore as error approach, is what I'm calling it. So that's when you just fit the model with and ignoring any differences in timing of year when the observations were taken. And there's kind of two forms of this. You can treat it as a continuous variable. So for example, July 1st, 2004 is about the middle of the year. So that would be 2004.5. Or you can treat it as an integer and just truncate it and just call it 1994. And so in the continuous version, growth is the same. It's constant all year long, from January through to December. And in the integer formulation, there's no growth all year. And then all of a sudden, all the growth occurs in an instant on midnight, December 31st. So these models aren't necessarily particularly realistic. So we wanted to see if we could formulate a model that would work a little bit better. So we wanted to be it to be an explicit statistical model that would be more realistic, that would relax the assumptions, particularly the assumption that the growing season is known without error. We wanted to quantify uncertainty, and so that uncertainty gets incorporated into parameter estimates and hypothesis tests and stuff further on. We wanted it to incorporate the growth observations that we're actually trying to analyze. So we wanted to fit it to data. We did not want it to be substantially more complex. We didn't want to have to estimate a whole bunch of additional parameters. And we did not want to have to use a bunch of additional data. So for example, we didn't want to have to use a model that required water temperature data throughout the study period and things like that. So in order to describe seasonal growth, we used telemetry on juvenile snapping turtles. So we chose juveniles because they're growing faster. Basically, it allows us to measure growth just within a single growing season. And we tracked 11 juveniles for one active season, and this was done in 2013 and 2014. And it's plotted there. And as you can see, there's a lot of variation in how much they grew, and that's because there were different sizes when we first started measuring them, or some of them were in different habitats, and of course, there's individual variation. And so the way we parameterized the model is we had one parameter for the amount of growth, and then the rest of the model described the proportion of that accrued at each time of year. And we called that the logistic growth phenology model. So phenology is timing of events or processes throughout the year. And this is a kind of a sigmoidal model, basically based on empirical results from the literature and also what we thought would best describe our data. So it starts, it increases gradually at the beginning and decreases gradually at the end of the active season. And it's based on a logistic curve. So it has two parameters. There's B, which is determines the midpoint, and G, which determines the rate. And so that determines how long the growing season is. And what you can see is, so for these fitted results, you might notice how short the 90% of the growth season is. And so it's basically 90% of the growth occurs between the end of May and the end of July. And I'll revisit that a little bit later. And when we compare it to the implicit models, you can see it's quite a bit different. So the growing season is quite a bit shorter. And it has a different shape. So the estimated growth is quite a bit different than what you would assume if you just assume that growth followed the active season exactly and that you knew the active season exactly. But of course, our objective is to combine this into overall lifetime growth model year over year, such as the von Bertalanffy. And this took a little bit to figure out, but the solution is actually pretty simple. We start off with linear time. And then on the y-axis, there's growth time, is what you can think of it as. And so that's just the proportion of growth expected to be accumulated by a particular time of year in that population. And we just used this growth time model, we applied it as a function to the time term in the von Bertalanffy. And so this could be applied any other growth model that you wanted to as well, like Gompertz or something like that. And when you plot that out, it would look like this, which may or may not be what you expected, but it looks a little bit weird, but it's a von Bertalanffy curve. And it has this kind of stair-step appearance because during a lot of the year, there is no growth occurring. And then there's these brief periods of fast growth in the active season. So this is just an example. It's just showing the curve for the median curve for male snapping turtles. We also wanted to be able to apply this model in a situation where we didn't have these kind of targeted specific within season sampling. So we looked at painted turtles and we looked at juvenile painted turtles. And that's just to keep the analysis a bit simpler because we didn't want to deal with maturation effects or sex effects. So we assembled a data set of 658 juvenile painted turtle observations taken between 1991 and 2017. And individuals in this data set were typically observed only once per year because the sampling design is basically focused on annual sampling. And so there's only 13 exceptions where an individual was observed more than once. But there's still some seasonal information in this data set. So just because they weren't always caught at the same time of year. And then we also did this analysis in a Bayesian framework, which means that we could specify informative priors. And this is an important part of our analysis, but I'm not going to talk about it very much now just for the sake of time. But you can see our paper or ask me questions after about that. So this is the result. This is shown for one example individual where we happen to know that individual's age. And it's shown by individual because this is an individually varying von Bertalanffy model. And you can sort of see there's like the little inset shows the individual's entire growth history, at least as long as it's juvenile. And close up, you can see how the model fit, and you can see the seasonal effect among years. And when we pull out specifically the seasonal components, you might notice that it has much wider credible intervals than we saw with the snapping turtles. That makes sense because we didn't have this repeated seasonal sampling. So there's a little less precision. And also the growing season is longer. And to compare, when we compare the two species directly, you can see that it is indeed longer. So for snapping turtles, 90% of the growth occurred in just 56 days. So that's less than 2 months. Whereas for painted turtles, their growing season was about 80% longer. And that's consistent with their with what we know about their thermal ecology and behavior ecology. So other research has shown that snapping turtles tend to be thermal conforming. Their body temperature is often lower than available environmental temperatures. And the other thing is, like, you know, we chose the active season based on what was stated in literature. And so, for example, May, snapping turtles have a lot of activity in May, but it turns out when you look at the telemetry studies, that's mostly males. And they're doing mate searching, and the females aren't very active, and that probably indicates there's not a lot of feeding and growing either during May. So that would explain why the snapping turtle growing season starts a little bit later. And painted turtles, by contrast, have higher indices of thermal exploitation. They put a lot of effort into thermal regulating over a longer period. So for example, like if you go right after ice out, you often see painted turtles like basking on the edge of the ice. And so they're more precise thermal regulators. And that's probably how they're able to have a longer growing season. So just to summarize, what we concluded for our growth model is that the logistic growth phenology model that we designed has 2 seasonal parameters, just like the implicit active season model. They both use prior knowledge of the growing season. But the implicit model uses only knowledge of the growing season, whereas the logistic growth phenology model is also fit growth observations, and it quantifies uncertainty, and it has a more realistic shape. Basically, it better matches the empirical observations of how ectotherms grow in a seasonal environment. Okay, so with that, I'd just like to quickly acknowledge Turtle Crew, all the volunteers, the field techs, and the other researchers at the Algonquin Wildlife Research Station that helped us out. Of course, the staff at the Algonquin WRS and Ontario Parks, funding agencies, NSERC, Laurentian University and the University of Guelph, and also Amanda down there with that huge snapping turtle. She helped draw the illustrations for that data analysis sausage diagram. So, even if there's time, I'll take some questions.
Hello, so David Lesbarrères and I live in Sudbury for now. Et ça me fait plaisir d'encadrer les deux dernières présentations du groupe de Yann Surget-Groba. On va commencer par Daphné Bernier qui va continuer les questions de génétique des populations de tortues mouchetées.
Parfait. Donc, bonjour tout le monde. Moi, c'est Daphné Bernier, candidate à la maîtrise en biologie sous la supervision de Yann Surget-Groba à l'Université du Québec en Outaouais. Et aujourd'hui, je vais vous présenter mon projet de maîtrise que j'ai tout juste de commencer sur la génétique des populations de tortues mouchetées. Donc, la génétique des populations, c'est des espèces en péril, puisque ces espèces-là sont souvent retrouvées en petites populations et des populations souvent fragmentées ou isolées, ce qui peut amener, dans le fond, une baisse du nombre de partenaires potentiels pour la reproduction et une augmentation du risque de la consanguinité. Puis tout ceci peut amener à une baisse de la diversité génétique qui est importante pour la résilience et la santé des populations. Dans le cas de la tortue mouchetée, vous voyez ici son aire de répartition. Et donc, comme Yann l'a mentionné, là, au Canada, c'est surtout dans le sud du Québec et de l'Ontario et une population en Nouvelle-Écosse. Puis, c'est divisé en deux unités de gestion selon le COSEPAC. Donc, la population de Nouvelle-Écosse est génétiquement différente et la population des Grands Lacs. Par contre, c'est considéré comme une grosse unité de gestion avec des sous-populations, dont celle de l'Outaouais. Puis, celle de l'Outaouais n'a pas été étudiée au niveau génétique et est considérée comme sensiblement pareil aux autres. Un des problèmes avec ça, c'est que ça pourrait être une unité indépendante. Puis, pardon. Dans ces cas-là, l'espace prend beaucoup de temps, de personnel et de ressources, comme Yann l'a mentionné, et donc on veut chercher une alternative. Dans le cas de mon projet de maîtrise, il y a deux volets. Le premier volet, c'est pour aller connaître l'état de la structure génétique et de la fragmentation, surtout au niveau des populations de l'Outaouais, mais aussi du côté de l'Ontario, au niveau de la ceinture de verdure d'Ottawa, pour identifier les populations qui seraient isolées et qui nécessiteraient une gestion en tant qu'unité indépendante. Le second volet est par rapport à l'ADN environnemental. Donc, c'est l'ADN que les organismes vivants laissent derrière eux dans leur environnement par les excréments, l'urine, le sang, etc. Et qui est possible d'aller récupérer sur un filtre pour ensuite analyser en laboratoire. Ce second volet de ma maîtrise va servir à évaluer si c'est possible de trouver la structure génétique seulement avec l'ADN environnemental, ce qui permettrait de réduire le dérangement des individus et des populations quand on va échantillonner et d'économiser temps et argent. Ce qu'on attend de ces volets-là, donc pour le premier volet, on s'attend à ce qu'il y ait plusieurs sous-populations en Outaouais qui soient plus ou moins connectées, voire complètement isolées, étant donné qu'on assume que la rivière des Outaouais constitue un obstacle difficilement franchissable pour ces petites tortues-là. Excusez-moi, j'ai mélangé avec la deuxième partie. Donc, que les populations de l'Ontario et de l'Outaouais soient isolées génétiquement en raison de la rivière. Puis, on s'attend que l'Outaouais soit fragmenté en raison de la topographie du terrain. Le volet 2, on s'attend qu'il y ait au moins une ou plusieurs combinaisons de microsatellites, donc des parties de l'ADN des tortues, qui permettraient l'évaluation de la structure génétique, tout en discriminant pour les autres espèces de tortues. Quand on a la tortue dans nos mains et qu'on prend un échantillon d'écailles, on sait quelle espèce c'est. Par contre, quand on utilise l'ADN, on a un pool génétique de tous les individus du milieu humide, donc plusieurs espèces de tortues. Et s'ils ont des ADN semblables, ça peut être plus difficile à identifier. Donc, on veut trouver une méthode qui va identifier seulement les tortues mouchetées. Donc, on a déjà commencé la prise de données. Donc, cet été, on est allé collecter de l'ADN dans 8 milieux humides de l'Outaouais. Vous pouvez voir ici la collecte d'ADN. Puis, dans les mêmes 8 milieux, on a fait de la capture, marquage, recapture. Et on a pris des bouts d'écailles pour analyser en laboratoire leur ADN. Puis, pour l'été 2022, on planifie faire le même protocole, mais du côté d'Ottawa pour pouvoir comparer les deux populations. Donc, un énorme merci à l'équipe de l'UQO, Yann, Laura et Émilie, à l'équipe du MFFP, de la Commission de la capitale nationale, Conservation Nature Canada et Environnement Changement climatique Canada pour leur soutien, sans qui le projet n'aurait pas pu avoir lieu. et en espérant vous voir dans les prochains congrès pour vous présenter mes résultats.
J'essaie de faire passer. On est tellement nombreux aujourd'hui à CHS que c'est dur de voir s'il y a des mains levées, mais j'en vois pas. Donc, on va passer à la présentation suivante, qui vient de la même équipe. Et cette fois, c'est Laura Désilé qui va présenter ses travaux.
Alors, bonjour tout le monde, je me présente, je m'appelle Laura Désilé et je suis étudiante aussi à la maîtrise à l'Université du Québec avec Yann Surget-Groba. Donc, aujourd'hui, je vais vous parler de mon projet sur la tortue mouchetée et de l'estimation de son abondance à partir de l'ADN environnemental. Donc, tout d'abord, la protection et la conservation d'une espèce menacée commence par connaître précisément son abondance et sa répartition géographique. L'abondance est une donnée très importante dans l'écologie car elle permet de savoir l'état général de la population et elle est essentielle pour la mise en place d'un plan de rétablissement. Les méthodes de suivi traditionnelles comme l'observation, la capture et le marquage sont parmi les méthodes les plus utilisées en écologie pour estimer la taille d'une population, mais ils demandent un grand effort d'échantillonnage et ils sont quand même très invasifs pour l'espèce. Pour améliorer la détection et le suivi des populations menacées, il serait avantageux de développer une nouvelle méthode d'échantillonnage plus rapide, plus sensible et moins invasive. L'ADN environnemental. Donc, c'est une méthode de plus en plus citée dans les recherches pour son faible coût et sa rapidité à détecter une espèce. L'ADN environnemental est une méthode qui se base sur la détection du patrimoine génétique de l'espèce pour l'identifier. Donc, pour récupérer l'ADN de l'espèce, il faut juste prélever un échantillon directement dans l'environnement. Donc, on prend un échantillon d'eau. Comme on peut voir sur les photos ici, il n'y a pas vraiment de contact entre nous et l'organisme, donc c'est très peu invasif. C'est vraiment un avantage de l'ADN environnemental. La méthode utilisée pour l'ADN environnemental, c'est de filtrer une certaine quantité d'eau, donc environ 2 litres, et de récupérer le plus d'ADN environnemental dans nos filtres. Après, cet ADN va être purifié en laboratoire et analysé par un test PCR pour détecter l'espèce cible. Donc, c'est une méthode quand même assez prometteuse, mais elle a surtout été utilisée pour détecter la présence d'espèces aquatiques et non pour estimer l'abondance. Donc, l'objectif de mon projet de maîtrise, c'est d'évaluer s'il est possible d'estimer l'abondance de la population de tortues mouchetées uniquement à partir de l'ADN environnemental, vu que c'est une espèce menacée au Québec. Ça serait très bien. Donc, nous, on est sortis cet été sur le terrain ce printemps. Donc, on a étudié 8 sites habités par la tortue mouchetée dans l'Outaouais au Québec. Sur chacun des sites, nous avons collecté 30 échantillons d'ADN et installé 30 verveux durant 9 nuits consécutives. Des données de capture ont été prises sur les tortues mouchetées capturées, comme le sexe, le poids et leur longueur. Elles ont été marquées, comme on peut voir ici la photo, une tortue mouchetée qui est en train de se faire marquer et relâchée par la suite. On a capturé au total 187 tortues mouchetées, donc ça a été quand même très fructueux, pour un total de 2160 nuits par piège. La prochaine étape de mon projet, en fait, ça va être d'analyser ces échantillons d'ADN qu'on a récupérés en laboratoire et de calculer un indice d'abondance de l'espèce à partir des données d'ADN détectées sur chacun des sites et de le comparer avec un indice d'abondance de l'espèce réel estimé à partir des données de capture. Donc, si on observe qu'il y a une bonne ressemblance entre ces deux abondances, donc on peut avoir une bonne corrélation entre les deux. Comme on peut voir, ça, c'est un exemple de graphique qu'on aimerait avoir d'une bonne corrélation. Si c'est là qu'on obtient, eh bien, on pourra déterminer, confirmer que l'ADN environnemental est une méthode capable de détecter, mais aussi d'estimer rapidement l'abondance de la population de tortues mouchetées. Alors, voilà. Merci beaucoup à tous ceux qui ont participé lors du terrain et merci à vous de m'avoir écouté.
Merci. J'ai une question pour Laura. Est-ce que vous avez cette idée de la saisonnalité de l'ADN environnemental? Je crois que peut-être il y a beaucoup plus dans l'eau dans le printemps, puis pas vraiment beaucoup lorsque la tortue va faire nesting season.
Oui, donc oui, en fait, on a pris ça en considération. On a échantillonné en mai, qui est à peu près la période où la tortue mouchetée est le plus active. Donc, on l'a pris en considération pour justement ne pas le faire pendant l'hiver où il n'y a pas vraiment de tortue, là. Il va y avoir moins d'ADN. Fait que, en mai, juin, c'est là qu'on est allé. C'est à peu près là où il va y avoir la saison des pontes. Fait que les mâles et les femelles sont vraiment actifs, fait qu'ils libèrent beaucoup d'ADN dans l'environnement. Ça répond à la question. Pour là,
On peut prendre la question de Gabrielle qu'elle a écrite dans le chat. Y a-t-il d'autres moyens de déterminer l'abondance sans ADN environnemental si ça ne fonctionne pas?
J'avoue qu'on n'a pas vraiment checké vu que c'est vraiment l'ADN. Peut-être que Yann peut mieux répondre.
Ouais, ben non, pas vraiment. C'est ça. C'est sûr que les inventaires visuels, disons, j'avais le terme en anglais là, marchent bien, mais ça dépend vraiment de la saison et c'est pas très précis. Donc la façon la plus précise, c'est vraiment le trappage et ça prend beaucoup de ressources. Et dans des populations à faible densité, là on a travaillé dans des populations quand même où il y en avait beaucoup, mais par exemple en Abitibi, ils ont passé des milliers d'heures de capture sans en attraper une seule. Donc ils savent qu'il y en a, mais il y en a tellement peu que c'est très très difficile à capturer. Donc vraiment, on espère que l'ADN sera efficace.
Jonathan?
Merci Laura. Daphné, une question peut-être pour vous deux. C'est quoi la probabilité de détection de tortues mouchetées avec votre méthode? Avez-vous estimé ça comme avec du occupancy modeling peut-être?
Laura, tu veux y aller?
En fait, oui, ben, ça, je t'ai muté, désolé. Ce que Yann, dans le fond, il nous a mentionné, c'est qu'avec à peu près 15 échantillons d'eau, c'est assez pour obtenir une détection. Par contre, pour l'abondance, ça va être encore à déterminer selon le projet de Laura. Je ne sais pas si Laura, tu veux ajouter quelque chose.
15 échantillons de 2 litres?
Ouais, ben, en fait, le volume dépend, là, c'est quand le filtre devient bouché plus rapidement, on en prend moins, là, c'est soit 5 minutes, soit 2 litres, ça dépend. Mais en fait, ça va dépendre justement dans le modèle on va essayer de développer un modèle basé sur, comme tu disais, les occupancy models. On va essayer d'utiliser un modèle comme ça qui va permettre justement d'estimer ces probabilités de détection. Là, ce que j'ai montré dans ma présentation, c'est vraiment sur des sites où on sait qu'il y en a et c'est juste des permutations, en fait. Là, c'était très simple. Mais en effet, on va essayer d'estimer la probabilité de capture. Il y a aussi, on n'en a pas parlé, mais il y a les faux positifs qui sont un problème. Donc, tous ces paramètres, on va essayer de les estimer.
Et on peut ajouter la dernière question qui vient de Patrick, qui lui se pose la question de savoir si une année va être suffisante pour faire toutes les estimations que vous voulez. Et notamment la comparaison entre CMR et eDNA. C'est pour Laura.
Ben nous, on a surtout une année là, pour la maîtrise, fait que ça va être à partir de ça, mais évidemment, si on avait plus de temps et d'argent, ben, on pourrait faire sur plusieurs années puis voir. Mais pour estimer l'abondance, nous, on va faire une saison de capture qu'on a faite en été pour, à partir de ces données-là, voir si en fait, déjà là, si le modèle marche, avant d'en faire plus. Je pense que c'est à peu près ça. Peut-être Yann, tu veux rajouter?
Oui, donc en fait, on a des données sur 2 années. Donc, on a des données, je crois, de 2019, mais qui étaient plus pour des… là, on n'a pas fait du trappage, c'était juste des suivis visuels. Donc, c'est moins précis, mais on a quand même des données et on a l'ADN. Donc, vraiment, cette année, on a mis un gros effort pour faire les deux en parallèle dans la même période. Donc, là, on a vraiment les données d'ADN. Et juste après, parce qu'on fait l'ADN avant pour ne pas trop perturber l'habitat quand on met les verveux et tout ça. Donc, juste après, on avait les données de capture. Donc, on espère que ça permettra de valider la méthode, là, de voir si on arrive à avoir une bonne corrélation entre les deux méthodes.
So Matt, you measured in-year juvenile growth rates, right? So you're measuring. So how do you account for the error in this? Because I mean, growth is so slow throughout the year, right?
Yeah. So that was that was why we did juveniles, because it actually is like a big issue.
Yeah,
it would be less of an it would be less of an issue actually if we'd done the study like further south with a longer growing season where they grow a lot faster. You would think, you'd think that there's not, it's fairly easy to measure like something like straight line carapace length with calipers. It turns out, if you have a snapping turtle that's like kicking and squirming, and it's like if a little flake comes off the carapace, like that makes a bit of a difference. And that's why actually when you look at the plot, like there's errors. you know, those errors show up. There's one that's, it's not super proud, but it's still on the plot, you know.
For sure.
It's just, it's something like it does, it's why you need a large enough sample. So we have about 60 observations there.
My next question was about like homogenizing juveniles as like a one standard growth rate. Because I know I saw growth for hatchlings directly post-emergence. So I mean, could the younger age classes be having a longer growing season, given they could acquire food, thermal resources? Like
Right. So that's possible. It's possible, but we didn't see it. So we didn't have many opportunities to actually measure it, but the size of individuals in the spring is about the same as in the fall. The only exception is like, I did look into the literature on this and I cite some of that in the paper. And one of the issues is like their shape is changing partly because like they're still unfurling from the egg and their shell is starting to harden up. And so there's not a lot of, there is like a literature on this. It kind of goes back and forth. My interpretation of it was that there was not a lot of growth to be expected in the fall, but we didn't actually make any assumptions about that. We just fit the model.